Oops. Good morning, Katarina. Good morning, friends. Good morning, Jamie.
Hello, everyone.
Hello.
How are you today, Victoria?
Hey, sorry, I'll make you moderator. I was trying to select the topics. Isn't there a rejuvenation or something like that here as a topic? I can't. Maybe house. Okay. How are you? Welcome, everyone.
Welcome, welcome, everyone. Okay.
Can you hear me?
Is rejuvenation
Oh, okay.
Yes, yep. Can hear you perfectly.
Perfect.
Did you say rejuvenation's a topic option in Clubhouse? That's a very specific topic option.
It's not, but they are very specific one for some areas. But it's not an option. How can we suggest topic options?
That's a really good idea. I'm going to do that too. Because, yeah, I mean, it's one thing to be creative with the topic options, but sometimes it would be nice to have a few more.
Yeah, right? Like more specific ones. Not just engineering in general, for example, or just biology in general. Like would be cool to have like more subtopics.
Even just to let you custom make your own ones.
Ooh. That.
Mm-hmm.
Because something I find is that sometimes the emoji that goes along with the topic word isn't the vibe I'm going for. And so
Yeah.
that would be really great to be able to make our...
Hi, Christoph. How are you today? Thanks for coming.
Hey,
Hi,
I'm fine, thank you.
Very pleased to meet you. I'm Jamie. Nice to meet you, sir.
Yeah, meet Jamie, meet Victoria.
Nice to meet you.
They will.
Hey,
Thank you very much for coming.
yeah, no worries.
And we'll wait like a few minutes until we start for real. And yeah, if that's okay with you.
Yeah, that's perfect.
What time is it? 6 hours later, right? 7 p.m.
Yeah, now it's 7, yeah.
Okay, cool.
Oh, where are you?
I'm in Zurich, in Switzerland.
Oh, so pretty. Wonderful. That's nice.
We're getting a lot of speakers in Switzerland lately.
Yeah, I heard the weather is improving. That the weather is nicer now.
We're all from different areas, Christoph. I'm in Scotland and Caterina's and
Oh,
you're in New York as well, aren't you, Victoria?
I'm in Oregon.
Oregon.
I'm
Victoria's
in
from Oregon.
Oregon. Yeah. I'm about to go outside because it's not raining.
okay.
Have you had a good day so far, Christoph?
Yeah, really busy right now because I'm so the project I'm presenting today was just part of my PhD, and so I'm gonna and I'm for postdoc I'm actually moving to Boston soon. So I'm still sort of finishing up stuff here, and then moving in July if everything works out.
Wow, this is only part of it. Wow. Wow.
Yeah, Boston is okay. I think to adapt from Europe to Boston is pretty easy. It feels like a North European city. I feel like.
Yeah. I guess I mean in terms of sizes, like everything in Switzerland is tiny, right? I mean, with a population of around 7 million, like It's still Boston is almost as big as Switzerland, I guess.
But
So
I think Boston feels like that. I think Boston feels like a smallest. I don't know.
yeah. I'm really curious how it's gonna turn out.
Which university are you going?
I'm going to the Whitehead Institute at MIT.
Nice.
Katarina, do you have an echo?
I don't know. I don't
Okay,
think
it's
so.
gone.
Yeah, it's gone.
Yeah.
You had some really crazy echo for a second there, but it's gone. The ghost has left the machine.
Sorry about that. Yeah, friends of mine used to live in Boston, but right now I think everyone left for their like assistant professor positions and stuff. But yeah, it will be Boston is pretty okay. It's not too stressful. The weather is not great. Like but anyway.
At the moment or generally?
Well, the winter is annoying, right? It's
Yeah.
But isn't there really good public transit there?
Boston, yeah.
Yeah.
It's like similar to Europe, I feel like.
That's why I think that I haven't been, but from what I understand, it seems like that would be something that would make it a smoother transition, is that in larger Northern European cities I've been to, there's such fantastic public transit. And so I think Boston has that as well. And the center of town is laid out with consideration to people actually using it.
Yeah, it's really good. I mean, here actually, like only few people actually own cars because the city also makes it hard to move around by car. So they want you to take public transport, and actually most people do it. So it's really nice to have everyone use it.
Yeah, I love that. I remember that of visiting Zurich.
I think can start in 1 minute, everyone. Thank you for coming. We'll start in around 1 minute, and yeah, this will be a really exciting. It's such a interesting and cool research. It's really exciting. So did you get a lot of like? Was this paid by rejuvenation type of grants or you?
Actually, this specific one was paid by Volkswagen. So
Oh wow!
there's like a foundation from them where they fund science, and this was one of those projects. But sort of now it's so as the project went on, it also went into a relatively big European grant, so funded by the EU, the ERC Advanced Grant. Where actually now we have quite a few people working sort of in the direction I was sort of starting with this particular project.
Interesting. That's so interesting. That VW. So were they mandated to have, or do they do this for tax break, or just in general they just do this?
The Volkswagen, I actually that like I'm actually not quite sure how the connection from the foundation to Volkswagen itself is going, but apparently at some point they just laid off some money that sort of was I don't know where the money from the company came from. And this pool of money sort of is maintained by this foundation that then uses it to fund science. But I'm actually not quite sure whether there's an actually control right now of the car company still over that pool of money, or whether it's sort of like a own type of foundation, maybe like Zuckerberg Chan Foundation or something. I mean, they fund single cell science, but it's not connected to Facebook anymore. So it's really sort of,
Well,
yeah, sort of more like that.
Volkswagen. Sorry, I didn't mean to cut you off.
Yeah, no, you
Yeah,
can
I
talk.
was going to say they have to make good on their name, because now they did that little bit of trickery in
Yeah, true.
diesel with the emissions, the nitrogen oxide emissions little thing, and then their name, saying they were going to change the name to Volkswagen. So I think maybe that's part of their
Yeah, although the foundation, I think, exists since like the 60s. So maybe then, maybe they did something bad then as well.
yeah.
But sort of the foundation is not sort of a reactive answer to that, I guess.
Okay, interesting. I think we can start. So, welcome everyone to the Science Society. We are very honored to have our guest speaker here today, talking about his recent research. And let me give you a little bit of information about Dr. Christoph Gäbelein. He did his bachelor degree in biology at the University of Freiburg, and his master's degree in chemistry also at the University of Freiburg. And later he did his PhD at the ETH Zurich, and his dissertation was about single cell engineering using fluid force microscopy. And this is basically this work, what we are, and based on the publication that we are listening today, is also based on his dissertation. He won also a few awards and grants, and yeah, we just heard that he will soon be working at MIT in Boston. So yeah, welcome, Christoph. And thank you so much for taking the time and speaking with us. And if you don't mind, Victoria will ask you first like a couple of general questions, and then the stage is yours for your presentation.
Thank you.
All right, so welcome. Science Society welcomes you, and freut mich, Christoph. We're so happy to have you here and excited to hear about your research. So before we begin, just to give maybe a bit of a human connection to who you are and what you've been doing, my question is, can you think, if you look back in your life, maybe even when you were a child, any time, when you noticed that you felt a spark that really interested you in science, that let you know that science was something that you felt connected with, and it could be a relative or a class or anywhere along your path?
Yeah, I guess actually, so after I finished school when I was like 18 or 19, I wasn't really good and interested in school because it sort of bored me a little bit. And then I started working like different jobs. So I worked as a postman, I worked as in sort of like a warehouse, and had to do civil services in a hospital. And sort of all of those jobs became too really repetitive relatively quick, and then that really bored me. So then I wanted to change jobs quickly again. And then at some point I started studying biology, and sort of at some point really sort of going more into sort of just scientific thinking, and always sort of inventing new concepts about stuff, just like really kept me going and fascinated me, and sort of just continues to amaze me, to sort of get to know new stuff every day. So that really keeps me in science and keeps me going. Yeah.
Thank you. Yeah. Oh, now I have the echo. It's catching. Okay, I hope it's gone. Yeah, it's so exciting to hear what our guests answer, because there is the thread of curiosity and following the curiosity, and so it's just wonderful to be around such passion and know that you're enjoying your work, and that's interesting. So maybe the civil service, the repetitiveness of that reinforced how important it is to be doing work that you love. And so my next question is, how did that work lead you to the path that you are on that brings you to the research that you're currently involved in?
Oh, that's a tough one. Actually, sort of, yeah. As you mentioned, I switched from biology to chemistry, and that was also something where sort of I wanted to get deeper into sort of new ways of viewing at science and viewing biology also, and that sort of led me to pursue more initially it was more more into the direction of biochemistry, and then from biochemistry actually I also switched again and did actually my master thesis in microsystems engineering. So I sort of keep sort of skimming over the sciences in order to find what I actually love. And then after going from biology to chemistry and then to engineering, I sort of ended up in my thesis here in Zurich in a discipline that sort of combines these three, where we do nanotechnology engineering in order to manipulate single cells. So it's a combination of going and going across sciences and really working on an interdisciplinary field.
So it's all beautiful that there's the echo again. That you're able to engage all three of your interests. And did you have a mentor? I'm just curious, as you're mentioning changing direction, were you so self-directed that you knew this is what you want to try and this is what you want to try, or do you feel that was that challenging to feel that you were doing the right thing for yourself?
Yeah, actually, so there wasn't much tactic involved. I always just picked whatever sort of caught my interest. I wouldn't say that there was a big part of a mentor or something. It's just like, we sort of I got into synthetic biology actually relatively early in doing in my bachelor's. And sort of synthetic biology is already sort of really cross disciplinary, and sort of for that point of view, I then drafted on, I guess, towards what I'm doing now. So I guess I don't have a really clear answer for that, but yeah, I guess I just go where I see something that really amazes me and sort of pushes me on.
And may I just say you're in good company. I'm echoing now. You're in good company because interdisciplinary topics is what we love the most in science society, isn't that right, Katerina?
Yes, definitely. Thank you. It's perfect. You're perfect for here for science society.
Thank you.
Those were great questions. I did the same thing. So I wasn't great in school either until I got really interested in something. Then, so yeah, thank you for answering those questions, and the stage is yours for your presentation. Thank you.
Okay, so again, thank you for your invitation and welcome to my presentation. So if you downloaded these slides, then right now would be the intro slide. So I'm part of of the group of Julia Vorholt, where we focus on developing nanotechnology for application in life sciences. And we work relatively closely together with the Department of Biomedical Engineering, where we sort of switch back and forth in between techniques. So the aim is really to sort of really develop techniques that actually sort of enable bridging the gap from actually just purely technology focused research to actually something that can be applied sort of on a more or less everyday basis by biologists or by life scientists. So we focus on the physical manipulation and sampling on the single cell level, meaning that we really directly interact with single cells with miniaturized probes we built. And mostly we are working with mammalian cells or eukaryotic cells in cell culture. We also have some projects now where we are working with fungi, but the main part is really on mammalian cells, I would say. So now I'm going to slide 2. So sort of the biggest challenge in our field is miniaturization, because sort of the volumes we deal with are just really, really small. So if you look at the size of a single cell that sort of make up the bulk of our body, their volume will be in the range of 2 to 5 picoliters, or mostly like smaller, so in the picoliter range, which is 10 to the power of minus 12 liters. So it's a really, really small volume. And sort of these spheres I plotted there sort of represent these volumes. And sort of if you look at sort of a milliliter, so a thousandth of a liter, then you can still it's still a volume you deal with every day. If you have a microliter, this every biologist or chemist maybe still knows from the lab is something you can manipulate just still by hand by using a pipette. Then if you go to another magnitude smaller, you're in the nanoliter range, where you have micro manipulators that can still deal with these volumes. And this is sort of the range where sort of these standard procedures end. So in the nanoliter ranges, you have micro injection devices that are perhaps, for example, used for in vitro fertilization of embryos. But those cells are really special by just being more than 1,000 times bigger than most cells in our body. And then if you're then one magnitude smaller again in the picoliter range, then you're actually in the range that is sort of relevant for biology and chemistry that is happening inside of single cells. Sort of those mammalian cells we deal with mostly have a volume of around 2 picoliters, so 2 to the power of minus 12 liters. And then if you go even smaller, then you're actually at the volume range that E. coli or subparts of the cell have. And the devices we built actually specialize in actually manipulating these super small volumes. And we try to also connect analysis platforms of these super small volumes. To sort of create output that actually can sort of we can make sense of. So creating data sets from single cells for through omics technology or by microscopy. And the tool we use to do this, so this is the next slide 3, is the fluidic force microscopy technology. And fluidic force microscopy is a technical platform that connects atomic force microscopy with microfluidics. And the centerpiece of this technology is the cantilever. And actually the cantilever, since I guess most of you on audio, is really, really tough to describe. So I looked it up on Wikipedia. And on Wikipedia, it says it's a rigid structural element that extends horizontally and is supported at only one end. So it's sort of, it's a super small structure that looks like an arm that you would just straight up hold out of your body. It's just really, really small. In our case, the cantilever is approximately cell sized, so its dimensions are the width is around 35 nanometers, the height is around micrometers. Sorry, 35 micrometers. The height is around 10 to 15 micrometers, and the length is around 200 micrometers. So this is the size that if you have a really, really small thing, this is just so you can just barely make it out by eye. So that you can just see that there is something, and this is the size of these probes we are working with. In our case, this is sort of the speciality of the cantilever. This probe is hollow, so so it's a hollow structure, and sort of the hollow inside of the structure we then later fill with fluids, and we use it as a microfluidic channel to manipulate these super small fluids we then actually extract or inject into individual cells. And on the apex of the cantilever, there's a downward facing structure that directly interacts with the sample. So we have a super small probe that we sort of hover over cells. And this super small probe at the apex has a structure at the end that we can directly push into our sample, which is, like, as I mentioned before, mostly mammalian cells. And this probe we actually then use to sort of extract parts of the cell or to inject fluids into individual cells. And the speciality of the atomic force microscope is that the probe can be positioned very precise in left and right, and it uses a force feedback mechanism to move the probe and to hold the probe in certain force ranges in that direction. But to explain that is maybe too much now. And in our case, the setup is also connected to a spinning disk confocal microscope, so we can sort of watch the manipulations we do in real time. And in sort of the discrete experiments, then our probe will hover over a culture dish in which we have the cells of choice that we would like to manipulate. And if you go on slide number 4, on the right side, you would see how such a probe looks on a scanning electron microscope copy image. So you can sort of see the size ranges of such an individual probe. And in our case, for this particular study, we designed a new probe type that, as sort of the connection piece between the microfluidic channel in the inside and sort of the cell, then later on, as we push the probe inside of the cell, has a cylinder that is sharpened on one side, so we can have sort of like a sharp syringe-like structure that we can push into individual cells. And in previous studies, this apex usually was actually sort of pyramid shaped, because it was just easier to produce. And really the crucial part of this technology is this cantilever, and within this cantilever, sort of the crucial part is the aperture, so the opening that actually then connects the fluidic channel to the inside of the cell as we push our probe inside of these cells. And sort of the size of the aperture determines the range of fluidic forces you then actually apply once you push the probe inside of the cell. And different structures inside of the cell, they require different aperture sizes, meaning they require different ranges of forces in order to be manipulated. And by tuning sort of this aperture size, so the opening of the microfluidic channel, we can tune which kinds of intracellular structures we actually manipulate once we apply negative or positive pressure to the microfluidic system. And sort of at an intermediate size, for example, you can sample just fluids from a cell containing endoplasmatic reticulum. And at higher sizes, you're also going to be able to manipulate mitochondria, which is sort of the topic of today. So after we manufactured and characterized these kinds of probes, in the first set of experiments, we went to fluorescently label mitochondria within U2OS cells. To manipulate mitochondria within mammalian cells is really an exciting and special thing. Also considering our background, because in the evolution of eukaryotic cells, mitochondria really take a special place within sort of the cell architecture. They are really at the sort of center heartpiece of cellular energy metabolism. So they're really important for creating ATP, which is sort of the energy currency for a cell, if you will, and also have other important cellular functions. But they are also really important for deciding cell fate or in sort of if cells transition in between different states. And they are also linked to other cellular signaling processes like apoptosis. And moreover, they are really special because they still contain their own genome. So in our cells, actually, there are two genomes. So there's the mitochondrial genome, and then there's sort of all of the rest of the DNA, which is inside of the nucleus. And the mitochondrial genome is actually something that is super conserved in humans and also actually across all eukaryotes. But as we have sort of CRISPR or talents or sort of certain techniques to manipulate DNA in the nucleus, it is really, really hard to manipulate mitochondrial DNA, mostly because as we only have one or two copies of DNA in the genome, as mitochondria stretch across or all over the place within mammalian or eukaryotic cells, they harbor thousands of genomes. So while you have only one or two copies of the nuclear genome, you're likely to have in every individual cells between I think 200 and 1,000 copies of the mitochondrial genome. So it's really something that is fascinating, and it's really hard to manipulate sort of this pool of mitochondrial DNA. Also because sort of the techniques we use to manipulate nuclear DNA, they don't really work in mitochondrial DNA. So you cannot as easily replace or introduce new variances into sort of into mitochondrial DNA or into mitochondrial genomes. And also sort of in our perspective, it's really interesting to sort of create the possibility to take a look at a single cell and within into the heterogeneity that is also there within this mitochondrial population that is within every individual cell. So that is why we're really interested to manipulate mitochondria in individual cells. And on this slide 5, you can actually see a U2S cell, which is just a bone osteosarcoma cell line, where I labeled, fluorescently labeled the whole mitochondria, actually the inside of mitochondria, with a fluorescent label. And as we push this probe into the individual cell and then give under pressure, also negative pressure to the microfluidic system, you can actually see that we remove parts of the mitochondrial network from one individual cell, where depending on where you push the probe inside of the cell, whether there are lots of mitochondria or only few, you can sort of either sample just few mitochondria from individual cells, or you can sample bigger chunks of the mitochondrial genome, which is sort of in the middle panel. And sort of we can, because sort of these cantilever probes, they are made out of silicon nitride, which is mostly see-through, so you can just see inside of the probe and see how much you sampled and what you sampled. So you can actually count and see and quantify after extracting part of the mitochondrial network how much or how many mitochondria you extracted. And then once we have those mitochondria sampled within the cantilever, we can actually just push out the sort of sampled liquid wherever. So we can just push it out into a sampling solution, which we then later on analyze via mass spectrometry or other downstream platforms. Or in this case, what we did in this paper, we can then transfer this mitochondrial content that we extracted from an individual cell. We can later on transfer this into different cells. One sort of novel aspect that popped up when I did these experiments first was that mitochondria are mostly organized in most cell types as sort of a network, which is sort of not sort of these textbook mitochondria where they are often like shown just like discrete spheres that are all over the cell, but mostly they actually organize into sort of a connected tubular network. But once you start sort of physically pulling on this mitochondrial network, they change their shape and they change it towards what is called a pearls on a string phenotype. So which is something that really looks like a pearl necklace under the microscope. Which is really curious, which made us really curious because you have a sort of force-induced transition of mitochondrial shape, and we could show that sort of this force-induced transition of a tubular mitochondrion into this pearls of a string phenotype, then also leads to recruitment of endogenous proteins that actually then sever these pearls into discrete spheres. And then these discrete spheres is what you can see inside of the cantilever. And this is actually really important because, as I mentioned earlier, mitochondria are really important for cell fate. So if you actually damage those mitochondria in terms of structure, the cell would most likely die because it's sort of a strong trigger of cell death or apoptosis. So we can sample these mitochondria from cells, and then in the first set of experiments we went, and this would be slide 6, we went and took these mitochondria and then directly injected them into recipient cells. So we effectively transplanted mitochondria from a cell type A into a cell type B, which is comparable somewhat to what you have in standard surgery, where you can take organs and put them into another person, just on a single cell level. When we do this in the beginning and we sort of label both mitochondrial networks, so we take a fluorescent label A, in this case red labeled mitochondria from sort of the donor cell type, and then push these mitochondria into recipient cell type that has differentially labeled mitochondria in this type blue, but it's shown as white. In the beginning, you can see that sort of the cell reacts a little bit stressed. And then in most cases, you can actually see that sort of as you have those two mitochondrial populations within an individual cell, you can see that these mitochondria then fuse with one another, which we termed to be mitochondrial acceptance. So the cell sort of recognizes these particles that we introduced as mitochondria and then fuses them to their own mitochondrial network. But this is not the case in all cases. So in few cases, actually, these new host cells, they actually also reject the mitochondrial content that came and then start to degrade them, which then results in sort of millions of really, really small particles that carry this fluorescent label that was inside of the mitochondria we initially transferred. In the beginning, we just did these kinds of experiments, just transplanting mitochondria from cancer cells to cancer cells. We could show that actually the procedure is really minimally invasive. So I guess in this case, it was over 90% of the cells actually then survive this procedure. So they're happy in all cases. We can monitor this uptake behavior in real time. And in this case, sort of the average number of mitochondria was around 6 individual particles, but there's sort of a spray of transferring only one mitochondrion to, in this case, I think the highest number was 15 individual mitochondria. In the sort of next set of experiments, and this would be slide number 7, we tested whether this also works on primary cells. And the reason being for that is that sort of there are mitochondrial quality control mechanisms in individual cells, and these quality control mechanisms actually don't work pretty well anymore in cancer cell types, or they degraded some of the key players of sort of the known mitochondrial quality control mechanisms. And if you then and primary cells still have active proteins that are part of this mitochondrial quality control machinery. So we use primary keratinocytes, which is skin cells that are sort of often also subjected to aging or DNA damage and also mitochondrial damage just by through UV radiation. And we transplanted mitochondria just from these labeled HeLa cancer cells into these primary keratinocytes. And then in the top panel, you have sort of a time lapse over 15 hours where you can see that in these really early stages, all of those mitochondria are spherical, and then over time, over 15 hours or 15 and a half hours, these mitochondria start to fuse to the mitochondrial network and start to fully label the mitochondrial network of these recipient cells. So again, we were really curious of why these mitochondria are actually still accepted by this particular cell type. And we analyzed this sort of process of mitochondrial uptake a little bit further. And which we saw in some of the cells, sort of the mitochondria that we transplanted were sort of rearranged, where we counted initially a certain number of mitochondria. So sort of in the graph shown below, I think it was initially like 12 mitochondria. And we saw that sort of if you just image these cells over time, what we could see that is sort of the mitochondrial content we put into individual cells was processed by the host cells and then sort of increased sort of the numbers of particles before these free particles you can see on the slide then actually went to fuse with the rest of the mitochondrial network. So there is some processing going on, but we're not quite sure yet of what these cells actually then do, or how this processing of transplanted mitochondria takes place. What we could show, though, that the state of mitochondria that is being transferred from a cell type A to cell type B doesn't really matter for this uptake process. So we treated the donor cell population where we extracted the mitochondria with different chemical treatments and drugs, and in all cases, those damaged mitochondria, the majority of them was still taken up in those recipient cells, even though sort of these drugs we use, are sort of these standard drugs that are used if you want to study DNA mitochondrial degradation. So they are sort of inducers of mitochondrial degradation in cell types. But if you actually just have a small population of these sort of mitochondria that would be normally degraded in their host cell type and you transfer them, then this new host cell will not necessarily start to degrade them. But it will usually wait for a rescue and then still sort of rescue this small mitochondrial subpopulation and integrate it into its own network. Sort of this was sort of these sort of really short-term observations we made when transplanting mitochondria, and on the longer term, it's really interesting to see whether the mitochondrial genomes that we sort of co-transplant with transplanting the mitochondrial mass into new cells, whether these genomes are actually also propagated within new cell types, or whether sort of these cell types have a preference for their own mitochondrial genomes and will sort of maybe take the proteins in the beginning, but then not continue to replicate the new types of mitochondrial DNA we entered into these particular cells. And we could show that if we use mitochondria from cell type A, in our case HeLa cells, and introduce these mitochondria into a population of recipient cells. Oh, sorry. In our case, we took mitochondria from U2S cells, put them into HeLa cells. And then we wanted to check whether we can still find the mitochondrial variances of these U2S cells in HeLa cells once those cells have grown for a couple of doublings. Actually, in our case, it was 2 weeks. And we could show that sort of depending on the amount of mitochondria that we transferred into new cells, these mitochondria or the mitochondrial genomes are actually maintained within the different cell types. So there's no inherent preference for mitochondrial genomes, at least not in the particular cell type, in the particular media we used. And we could do this because mitochondria do have certain variances, and so they have single nucleotide polymorphism, so their DNA differs, but only just a little bit in individual positions. And you can use those to actually then distinguish on a very high resolution the content of the genomic content of mitochondria of individual cells. So this brings me to the end of my talk. If you have any questions or something, please say something. Otherwise, I can of course tell you to sort of read the paper, because this is just a small show over what I did in this particular project. Thank you very much for listening.
Yeah, thank you for this amazing presentation. It's such an interesting work, and I shared a little bit in the chat what implications your work has, what it could be applied for in the future. Are you planning, like, will your next work be still related to this, or do you want
Yes, somewhat. So we still have. So there have been several publications relatively recently on sort of mitochondrial transfer actually happening in tissues in certain conditions. So where sort of cells rescue other cells via giving them some good mitochondria in order to sort of rescue their mitochondrial pool. And this is really something that's really interesting, and it's sort of really hard to sort of properly these studies to properly reinvestigate these kinds of occurrences, because sort of there's no other proper natural way in order to transplant mitochondria in between individual cells. Right now, I have two more projects on this particular topic on sort of cancer cells influencing other cells by transferring mitochondria, and the other one is on mitochondrial quality control, which are still very directly related to this topic. And sort of as a group, coming from microbiology, we are more interested in sort of the endosymbiotic history of mitochondria. So we're trying to learn to understand how sort of endosymbionts came into place, and how endosymbiosis work, and how sort of how do bacteria become organelles. Sort of this transition is what we're working on right now, and therefore we apply this technique as well to sort of manipulate mitochondria and to manipulate bacteria or endosymbionts that are also sort of mitochondrial sized within eukaryotic cells or within mammalian cell systems also.
That's so interesting. So would you know how would it be okay to transfer mitochondria, a different mitochondria type, into a human cell, like from another human that might be different? That would be one question. And then, like, if there's some sort of type of rejection response, if one would do so. And then the other thing I think is interesting, that mitochondria are so highly conserved, but they have a quite high mutation rate throughout an organism's or a cell life. Um, are you looking also into, since you mentioned like you're interested in like the evolution of mitochondria, are you also looking into how that is? Because it could give us insights into, rejuvenation technology, maybe.
Yeah. So actually, this is this is also something we are looking for in the future. There's still so many open questions in sort of cell biology of how actually cells are able to select for good or bad mitochondria. I guess this is sort of the main question we're also working on, because depending on cell type and cell state, there are some selection procedures known for sort of good or bad mitochondria, actually mostly in the germline. So for embryos or in Drosophila, actually, it is known that there are selection processes where cells are able to select for good mitochondrial genome variances. But in somatic cells, and in this study, we worked mostly with somatic cells, it's actually not known whether this is just a purely random process where you have mutation and you have propagation, and then these cells just go on and on and on. And if you're unlucky, sort of you catch sort of a bad population of mitochondria, or whether there are some cleansing processes, and this is really not known. There are mitochondrial, there are diseases associated with mitochondrial, with sort of diseased mitochondrial genomes, or sort of with DNA mitochondrial genome variances that sort of cause these diseases, and sort of finding out how actually sort of this cleansing process works in the germline could then help to activate these processes in patients that do have problems with their mitochondrial content. So this is sort of the direction of really finding out how stuff is working.
Yeah, sorry for taking so many questions, but that's really interesting, because my theory is for mental health, in neuroscience, that almost everything is maybe mitochondria related, and how chronic inflammation with like bad ATP production, like with high pollution, can lead to all kinds of disorders, especially in the brain where so much energy is needed all the time. So I think an interesting evolutionary question is that I have, and I don't have any funding or any ways to study, is with changes in having less scarcity in developed countries for generations now, right? We never had any animal living in conditions where you don't have to worry about food all your life for generations, probably. If that threshold changes, do the germline cells become more picky? Like, do fertilization processes become more picky because we have such a big abundance of resources that we the threshold for turning into an embryo will be higher? Is there any indication for that?
Sort of across humans, it's really tough, or let's say across mammals, it's really tough to judge, because sort of the mitochondrial genes and the mitochondrial gene content within mammals is really conserved. So it's always those 13 proteins it's still coding for. I think actually, if you and sort of, but if you look across different eukaryotes, for example, in yeast, yeast only has 9 mitochondrial genes. So across eukaryotes, sort of the genome, there are really large changes in mitochondrial genome content. So, or sort of plants still have plants really have less problems with energy, and their mitochondrial genomes are also way bigger. So there is sort of a selection pressure on that. But sort of even given that we always are really well supplied with food, I wouldn't say that that this is the main driver for selection of on mitochondrial genomes, even on a single cell level, because even as you have a lot of food, this can still be a problem for certain cell types, because you always need active mitochondria. Even if you have super high glucose all the time, you still need active mitochondria in order to produce amino acids, in order to have the cells growing.
So my theory is that it becomes more picky, so more fine-tuned, like, because we only have, like, it takes forever to grow into a full-term human. It's
Yeah,
very
yeah, yeah.
expensive for a female. So I thought maybe the more resources one has, the more pickier the system can become. And with
Okay, yeah.
that, having more infertility with certain partners, but then with other partners it works pretty well. And I thought maybe the system is just becoming pickier because it has the abundancy to spread out
Yeah.
So that's just my personal theory. So,
This is a super interesting theory, but it's really totally dark matter. It's really, really, really unknown. It is known that there are partner mismatches where mitochondria always come from the mother. So in all organisms, so you actually all the mitochondria you inherit, they always come from the mom, and usually there are no mitochondria coming from the father. And there are known cases where the mitochondrial genome of the mother actually doesn't fit the sort of nuclear genome of the father, and then you actually have couples that are unable to get children because of that. But how that mechanism works is really totally unknown. There's really no one knows how sort of the crosstalk between nuclear genome and mitochondrial genome actually works, and what sort of the selection pressure there is.
yeah, that's why it's so okay. Sorry for taking so many questions. Everyone, please go ahead, flash your mics and ask your question. Thank you.
Okay, I'll go first. First, Dr. Christopher, thank you so much for your fascinating talk. I had a few questions. What is the reproduction rate and the error correction rate of mitochondria, and HeLa cells? Why are they the ideal cell? This is a very common cell that's used in these sorts of experiments. And also, when you brought up the matrilineal passage of mitochondria, I was curious how that works in hermaphroditic organisms such as zebrafish.
Okay, I guess you have to. I'm gonna ask later for more questions. Sort of the reproduction rate of mitochondrial genomes is that's a really tough one. To be honest, I'm not quite sure. It's known that of course once per cell division, sort of the mitochondrial genome is sort of reproduced once, but sort of the turnover of all the mitochondrial genomes, to my knowledge, is not really known how high actually the turnover rate is if you look at an individual cell. Also because sort of now there's a theory that there are sort of diverse subpopulations of mitochondrial genomes where some of them are replicated more than others. So to my knowledge, this is not really well known. It is known that there's heterogeneity within the replication rate, but yeah, to my knowledge, on a single cell level, this is really unknown. It is also not known how actually the number of mitochondrial genomes per cell is regulated. So if you remove or deplete mitochondrial genomes within individual cell types, which you can do by drugs, by adding some drugs, the cell afterwards will go back to its original state with the same number of genomes. But it's not known how this type of regulation works. Sorry, could you say your second question again?
Sure. The second part was what's the error correction rate, and then the second question itself was why are HeLa cells ideal for these types of experiments?
Okay. Sort of the error correction rate, it's out there, but I'm actually not quite sure. I know that the mutation rate is higher. Sort of mitochondria have their own polymerase, but and its error correction rate, I think, is 10 times higher than. So no, the error rate is 10 times higher than the one from the nuclear polymerase. So the average error rate within mitochondria is higher, and also the mutation rate is higher, likely because they are reactive oxygen species. In our study, we work with HeLa cells just because they are sort of easy to work with. That's also why we just used them as a in sort of the initial experiments when we just tried to see whether we can actually make it work to extract and transplant mitochondria from individual cells. But for actually studying mitochondrial mitochondria or mitochondrial dynamics, they're totally not ideal. Mostly because they lack parkin, and parkin is associated to Parkinson's disease, and it's associated to Parkinson's disease because it has some way of controlling mitochondrial quality. And this mechanism doesn't work anymore in HeLa cells. So initially, we really just worked them because they're sort of easy to hold lab pets, and that's also why we then later on moved to primary keratinocytes, because they're sort of a more relevant cell type. And then the last question was something on Daniel Lerio. Could you ask this one again?
Sure. Knowing that mitochondrial mitochondria is the way that it's passed on, how does that work in organisms that are hermaphroditic, such as zebrafish and other organisms?
You mean in between different generations?
Yeah.
So from okay. In zebrafish, I'm actually not quite sure how the passing on there. I know for human, mouse, and Drosophila and yeast, and in all of them, there is a selection mechanism that makes sure that only one of the mitochondrial populations survives. So in humans and mice and Drosophila, it's always the female mitochondrial populations that will make it to the offspring. So that's why I guess it's the same in zebrafish. And in yeast, yeast cells fuse, and then if you have whole cell fusion, obviously initially you have both pools of mitochondria together within an individual cell, and then once you grow those yeast cells again after fusion, they have some way of separating their mitochondrial genomes again, and then sort of the offsprings will only have either the mitochondrial genome of sort of the previously fused cell type A or cell type B, but never mixed populations. But how this is working, no one knows to my knowledge.
More mystery.
Yeah.
That's the beautiful thing about science, right? The more questions you ask, the more answers you find, and then more questions you have, even more.
yeah. But those are really tough to answer questions.
That was great. Thank you. Can we see a flash of mics? Who else has a question? I see Dr. Shaw and LT. Welcome. Okay, LT, I saw your mic flash first, and then we'll pass to Dr. Shaw next. Go ahead.
Okay, thank you, Chris. My question is you when you in your slide number 8, you made like a few points. The last point, you were saying that the FluidFM methods extracted mitochondria is more effective than the bulk extracted mitochondria.
Okay.
Yes, if you recall.
Sorry. I skimmed over that point. So
you skimmed that.
Okay. I'm sorry. It's something I should have mentioned. But of course, there are also protocols where you can just purify mitochondria from individual cells, and then you just have pure mitochondrial extracts. And of course, we can also just put these pure mitochondrial extracts into the microfluidic channel of our probes, and then directly inject previously like sort of bulk purified mitochondria from individual cells, and then just directly inject these into individual cells, which sort of creates a higher throughput. But we found that sort of the extraction protocols that you have to use in order to purify mitochondria from sort of a bulk population of individual cells often leads to sort of a damage of the outer membrane of those mitochondria. And we didn't really find a protocol where we have like the super perfectly structurally preserved mitochondria after this purification protocol. And this leads to the fact that if we inject those mitochondria that we bulk purified into individual cell types, then the uptake rate is lower compared to the direct extraction of individual cells. So if we directly extract them with FluidFM and then transfer them directly within a minute or so into a new cell, so if the uptake rate and the maintenance of mitochondrial genomes is higher compared to using mitochondrial extracts.
Okay, thank you. So it's more the unscratched,
Yeah.
more like a complete, it's better. Thank you. That's it. Thank you so much.
We really appreciate these questions. All the questions just make the room better, and they allow us to really learn even more about what you've come here to share, Christoph. Thank you. Dr. Shah, the mic is yours.
Thank you so much, Victoria. So thank you, Christoph. That was a wonderful, presentation. My question is about the cancer because you mentioned about it, and we know that one of the challenges in immunotherapy specifically is the mitochondrial, reaction to the immunotherapy, and we have a different approaches, as you know, that such as checkpoint inhibitor, and CAR T cell specifically is my question. If you have further information around that, because specifically I was wondering about the mitochondrial pyruvate carrier and this process. Do you have any further information, or did you notice anything during the transplanting of the mitochondria? Does it impact it to this cycle or not?
To sort of you mean to recycle? Sorry, to
MPC cycle,
the cells.
mitochondrial pyruvate carrier.
Okay.
Have you heard about that?
Yeah, I heard about it.
Of the,
Yeah. So this is actually we're not quite there yet. We are trying to actually couple mitochondrial transplantation towards analytic tools of cell metabolism to sort of see how much of an impact actually mitochondrial transplantation has on sort of the energy state of individual cells, and whether this actually makes a difference or not. Also because sort of we have those publications where people describe that mitochondrial transfer takes place in nature. But this is also something we're working on right now. So we're not quite there yet. So maybe in a year or two I can give an answer on that.
Sure. And based upon whatever you shared with us, your experiment was include one time, right? You didn't, for example, apply multiple times of the regimen over the mitochondrial after transplantation, right?
So what is,
for example,
after
transplantation, I was just wondering, did you add this experiment of for example, for the radiation therapy, for checking of the quality or such a thing? You didn't have couple of the cycles, or you just how you check that?
Sort of the with the drug treatments, the experiments, you mean?
Yes.
Sort of we always repeated each experiment 3 times, and sort of the way we evaluated this was always we made a transplantation, and then we did time-lapse microscopy on these individual cells after the treatment. So in our case, the treatment was CCCP, which is an uncoupler, doxycycline, which is a drug that acts on mitochondrial genomes, and then H2O2, just as a general sort of radical and damaging mitochondria. And in each of those experiments, we sort of treated the donor cell population with these different kinds of drugs or agents for different amount of times, whatever was reported in the literature, which was sufficient in order to trigger cellular responses towards mitochondrial degradation. And then we started extracting those mitochondria and put them into the keratinocytes. And then in each of those 3 replicates we did for each experiments, we then followed each individual cell and checked whether it was able to, or whether how it would sort of act in answer to receiving these types of damaged mitochondria. Whether it would directly fuse them to their own mitochondrial network, whether it would degrade them, or whether it would sort of rearrange and then fuse them. So our readout in that case was always just optical microscopy. We are working now actually on using as I said before, direct metabolic couplers in order to see how much or how fast they are able to recover once they are inside of those cells, or whether they recover first and then fuse, or whether fusion itself is sort of the recovery process there. Does this answer your question?
Sure. 3 times same there. Thank you.
Jamie,
And,
then
and
Oh, I was going to say, yeah, Jamie, we haven't heard from you yet.
yeah, I was going to say thank you very, very much for your talk, Doctor. That was incredibly interesting. First of all, my question is, I was looking at your paper, and when you wrote in your paper that fusion with the mitochondrial network of recipient cells, you said it occurred 20 minutes after transplantation.
Yeah.
I found myself wondering, why 20 minutes? Is there some kind of thing that happens in the cell that it, I don't know, checks every 20 minutes to make sure something's missing? Or is there something else deeper happening that takes 20 minutes to begin this process of fusion? Or
so why exactly it was 20 minutes is tough to tell. What is known is that mitochondrial fusion requires connection of the mitochondria to the cytoskeleton. And then it also requires certain proteins to attach to the outer surface of mitochondria. So my guess is that once we remove those mitochondria, obviously from the cytoskeleton, from an individual cell, and then they are still surrounded by parts of the cytoplasm from the original cell. There is just some time frame in which it takes to sort of link these mitochondria then to the cytoskeleton or sort of the fusion structures that will then lead to fusion within the recipient cell. I just think that sort of that process takes some time. It could also be that sort of since we are actively pushing a physical probe into the cell, that sort of stresses the cell in the beginning and then sort of just takes some time to get going again. But yeah, usually sort of the transplant, once we put it into the recipient cell, it really takes some time in which you don't see the mitochondria move at all. So they really stick to the place, and sort of in the formation you individually put them into an individual cell, and then at some point they start moving and fusing. So sort of, and once they start to move, then they will also fuse relatively rapidly. So I think that's why I think that sort of this connection to the cytoskeleton just takes some time.
that's interesting. And is that, since I've got no frame of reference, is that considered quite quick, or for the cells to behave this way, or is that quite slow? Like, did you have any expectation, or did you just put it in there and just cross your fingers and hope that it would connect, would fuse?
No, to be honest, we didn't have any expectation. In the beginning, we thought that probably most of the cells will die because sort of mitochondria contain cytochrome C. And once mitochondria are damaged, this cytochrome C might leak into the cytoplasm. And cytochrome C is sort of a really known strong agent causing apoptosis. So actually we thought that probably we would damage the mitochondria too much during our extraction process, and then it would always kill the recipient cells. So that they fused at all was a surprise, actually. And then the rest is just descriptive. So we didn't have any expectations, but perhaps, of course, messing with protein levels that manage mitochondrial fusion might have an impact on the velocity of this uptake process. But we didn't test this yet. Yeah.
That's interesting. Sorry, another question I have is when you were saying before that mitochondrial DNA is prone to having disease-causing mutations, I was thinking, since I don't really know this field very well or anything, so please forgive me for the silly question, but is this similar to what they say about our normal cells when they replicate over and over time? They give way to mutation. That's when we get cancers and things like that. Is this the same principle with the mitochondrial DNA? Like when it's replicating itself, is it the same phenomena?
I guess it's the same phenomenon. So sort of, yeah. Since sort of the DNA polymerase introduces errors, the more replication or more cell replication would also likely lead to a higher likelihood of having sort of disease phenotypes in mitochondria. But since you have so many copies of mitochondrial genomes, it's not like one or two sort of replications going wrong actually makes a difference. So it is known that if you have sort of a disease-causing mutational pattern within your mitochondrial genomes, oftentimes like 60% of the population, so let's say 600 out of 1,000 mitochondrial genomes within an individual cell actually need to have this particular mutations in order to give rise to a diseased state phenotype. While if you only have 40% of mitochondrial genomes damaged, then you won't get the disease phenotype. So it's not like this 1-0 response you would have from cancer cells or from other cell types. So it's a much more sort of mild selection process, and oftentimes these since they are inherited from mother to daughter. Oftentimes you can have these things in a family, and then just stochastically one or two family members might get it and others won't, just out of pure sort of stochastic randomness and distribution of how mitochondrial genomes are inherited and are damaged over time. Yeah.
Wow. And another one, please, if that's okay. I saw it, and I hadn't heard of this before, but you were speaking in your paper about an atomic force microscope. I hadn't actually heard of this before. Is that as cool as it sounds?
Okay. Yeah, sort of the whole technology is based on atomic force microscopy. And atomic force microscopy is sort of a technique that actually was invented, I think, back in the 80s. And originally, this technique is really used to resolve nanostructures sort of in soft lithography or in chip production, or actually it is also used sort of to image single atoms. So these were sort of the roots of these techniques of really having super high resolution of structures, also in high vacuum. And then after some time, this particular atomic force microscopy platform then sort of transitioned also into life sciences. So yeah, the basis of our technique, so the machine we use is sort of a rebuilt atomic force microscope, which still sort of offers the resolution in principle to create really high resolution sort of force profiles of the cells we manipulate, and we need this technique actually to sort of enter the probe in a force into those cells that doesn't kill them. So if you imagine having something macroscopic, like a pipette and trying to push this into an individual cell. In all cases, you would kill it. And that's why we need this atomic force microscopy to actually get really good positioning and hold a still position within individual cells during our manipulation steps. Yeah.
That is amazing. Oh, thank you so much. I actually have one last one. I'll drop it really quickly so I don't take up all the stage time. But given the technology thing, you mentioned in the paper as well, fabrication of tips with customized aperture areas, right? Was
Yeah.
this something that you had to talk to specific engineers about to make this specifically for this kind of testing, or were these developed for other things somewhere else? Were you just catching up to see what engineers have and using it?
No, so we're working directly with a company that produces these AFM cantilevers. It's called SmartTip in the Netherlands, because the platform we're using is also commercialized, and with them we develop these kinds of probes. And the probe processing as it's going now is actually that they sort of produce somewhat prototypes, so sort of raw molds of these probes we're using, and then if they produce it on sort of a wafer-based scale. These are sort of techniques that are used usually for computer chip production. Mostly, sort of, it's sort of the same technological background. But some of the structures, some of the nuances, they actually can't produce because, it just has certain constraints of producing these structures. And that's where we actually step in and sort of taking further modification steps on those structures. And what we do is sort of destructive manufacturing, where you have a certain structure. In our case, this cantilever, if you want to see it, it's on slide 4. You have a cantilever, and it's actually sort of the aperture, sort of actually the side that connects the fluidics to the outside. This part always needs to be relatively sharp, and in order to make it sharp, we cut away parts using focused ion beam, which is a technology where you use gallium ions and destructively just can remove parts of the cantilevers by shooting at it with gallium ions. So it's called focused ion beam. Which is always coupled with electron microscopy. So it's sort of a special area of manufacturing to actually make it work to produce those probes. Yeah.
That is amazing. Thank you very much for indulging my questions, and thank you very much for your talk.
And Christoph, we've had you here over an hour. I see that we have Tamar and Eli on stage. Is it okay? Oh, sorry, I'm walking by a lawnmower. If they ask you a question, or do we need to say
for
goodbye?
Sure. I mean, no, I think 1 or 2 more questions are fine. Yeah.
So we've got 1 or 2 more questions. Tamar, welcome to the stage. Do you have a question?
Oh, thank you. I just took the end of the talk, but yes, I think when we think about mitochondria, we think about the age, the living age. This will interfere somehow.
Well, yeah. I mean, there are some relations to this, because there's sort of there's this whole mitochondrial hypothesis of aging that sort of, as there's no active selection process for mitochondria in our cells that we know of, at least, there is a hypothesis that sort of all aging happens because sort of the pools of mitochondria actually degrade within individual cells. And then, of course, if you actually enter sort of fresh or young mitochondria into individual cells, that this would sort of have positive feedback for the rest of the cell and sort of rejuvenate these individual cells. yeah, this is actually something, as I said, we are also interested in how sort of feedback of metabolic state of mitochondria and how this then relates to the state of the whole cell. So it's definitely sort of a relevant question. We're just not quite there yet. So there is a hypothesis. There are sort of indications that this sort of is happening within cells, but how the exact mechanisms are is actually unknown. We just know that it's related to the quality of mitochondria and of mitochondrial genomes, but it's not like a direct on-off thing. It is just known that if you have bad qualities and sort of high mutation rate of mitochondria, then sort of the aging process is sped up drastically. But how this is actually working is still a big unknown.
Tamar,
Really interesting.
sorry, go ahead.
No, I just appreciate it's really interesting research. Thank you.
Fantastic question. Thank you, Tamar. And so, Christoph, we have one more. I believe, Eli, we haven't heard from you today.
Yeah, hi. Sorry I joined
Hello.
late. Sounds pretty fascinating. So quick comment. I actually did do atomic force microscopy and scanning tunneling microscopy for a number of years, and I'll just mention that AFM in vacuum very challenging. At least to get single atom resolution. That only happened, if I remember correctly, in like 2015 or 2016 fluorine atoms or fluorine molecules on a surface. They actually did manage to resolve the single bonds, which is truly amazing. It's much more challenging than it would seem, but it had been used for cell imaging in the 90s. I'll spare everybody a particular situation or account of my cells being looked at by an expert in hematology. So what I wanted to ask was, are you considering or are you aware of any similar work on chloroplasts, especially looking at the effects, ways of improving crops by introducing chloroplasts from different organisms into, say, staple crops?
So actually we had trials on trying to manipulate organisms that harbor chloroplasts. In our case, this mostly didn't work because those cells are really fragile, and sort of all plant cells have a rigid cell wall on the outside, and once you make a hole in the cell wall, it's those cells usually die relatively rapidly. Whether actually sort of the thing is in crop yield is that usually I think the chloroplast quality is not sort of the thing that is missing. So because plant cells get their ATP from chloroplasts through photosynthesis, but usually they really have lots of ATP all the time because photosynthesis just works so well. What I know that sort of is limiting for them is actually nitrogen creating nitrogen and getting enough phosphorus. And there are lots of attempts to try to engineer plants that can fix their own nitrogen. So I mean, 80% of the air is nitrogen, but fixing it actually doesn't. This is only being done by some few bacteria in the soil. And there are efforts really to try to engineer plant cells in order to get these traits from those bacteria. But for crop yield, I guess sort of chloroplasts or chloroplast quality, at least I think it's is not sort of the limiting factor. So I don't know of engineering approaches that sort of went into that direction, but rather into the nitrogen fixation. problem.
Yeah, I had been looking at efforts that are being made to get cyanobacteria that fix nitrogen to become endosymbionts. It's interesting work.
Exactly. Yeah. But yet, to my knowledge, it doesn't work yet. So, but there are really huge efforts in order to get this done.
Is there a way to make a perforated patch like whole cell patch clamp recordings? Like,
Sorry, I didn't understand you acoustically. Could you repeat the question?
yeah, sure. Is there a way, like in whole cell patch clamp recording, we have the alternative to use a perforated patch? So instead of rupturing or punctuating the membrane physically or mechanistically, you use a compound that makes little pores that you can prolong basically the lifetime of that connection. Is there something like that maybe for plant cells? I don't know, Eli.
To create pores. Yeah. I mean, yeah. The problem is really the cell wall. Usually, to my knowledge, at least, that where actually, yeah, you have this rigid cell wall around cells, and then you have that problem. You can remove it, but then those cells are really fragile and die fast. So, yeah, it's really could be, but
Yeah, neurons use gramicidine, so it makes like we have to wait 15 minutes, makes little pores.
yeah.
You can some ions, but yeah, I don't know. Idea. Thank you. Yeah, I think you answered a lot of questions. We took up a lot of your time, and but thank you so much. This is such an interesting research topic, and your presentation was really great and great to follow. And yeah, it's so interesting. So I think your work will have a lot of applications and a lot of different disease models, and also rejuvenation and all kinds of stuff. So it's really important, and I wish you all the funding and the least bureaucratic hurdles and a smooth transition to Boston. So
Yeah, fingers crossed.
yeah, we'll be fine. And yeah, thank you everyone for coming and asking all these questions to make this discussion really interesting. And yeah, thank you so much, everyone. And if you like discussions like this, follow the club. We'll have more interesting talks coming up. We'll have actually later today Dr. Uyin for talking about building complexity in biological design spaces. He's at the university at CUNY. It's a new institute. He's the director there for nano engineering, so it will be really interesting. And tomorrow we have a dogs room. How dogs recognize dog and human emotions. Usually, species don't really care about the emotions of other species, but dogs are really good at recognizing human emotions, and it's a talk about that. And on Saturday we'll have Dr. Chang. She will talk about how she uses molecular orbital based machine learning. It's a really interesting paper. So yeah, thank you everyone, and a special thanks to you, Christoph, and good luck for everything.
Thank you. Thanks
And
for the
For your time,
invitation.
Christoph, and good
come
luck in
back
Boston.
next time.
Yeah, good luck in Boston. And
Vielen
yeah, maybe you'll want to come back again. If you listen to any replays, you'll see we have collective curiosity. We can keep you here for a really long time if you'd like.
Yeah, I agree. Yeah, thank you everyone. Hopefully you come back later. That will be a really interesting talk. And yeah, thank you. Hear you all soon. Bye everyone.
Bye,
Bye.
everyone. Thanks,
Thank
Welcome.
Katerina.
you.
Bye bye.
Bye,
Three,
See you all
Victoria.
soon.
one,
Bye,
bye.
Krista.